Thank you. So, as Jeremy pointed out, this is another talk or another project that has been carried out within the framework of the ABIR consortium. And I'm going to focus on genetic factors that contribute to the development of binding and neutralizing antibodies against interferon-beta treatment. So maybe let me first point out, so this is an ongoing project. So what I'm going to present to you now is just the current state of the analysis, and some minor details might still change at some time. So I'm presenting you what we are confident with at the moment. Interferon beta is a commonly used treatment against multiple sclerosis. And there are three major products, Avonex, Rebif, and Betaferon, which are different subtypes of interferon beta raised in different cells and which are also administered in different ways. Up to 40% of the patients who receive this treatment develop antibodies, ADA, against them. So if these antibodies just bind the drug, we call them binding ADA, BADA. And if they really interfere with the binding of the interferon to its receptor and thus with the biological activity of the drug, then we call them neutralizing antibodies, NADA. Okay, it has already been shown that genetic factors contribute to the development of these antibodies, and there's actually been quite a number of studies on this topic, but unfortunately there's no real consensus of what the genetic factors are in the end. Most of these studies focused on class 2 HLA alleles, and it could be population differences, treatment differences, whatever. What we wanted to do is to analyze a large sample, decidedly from two different sites, from the Karolinska Institute in Stockholm and from the Technical University in Munich, and see whether we can find some kind of consensus on the genetic factors in this sample. In order to make it as homogeneous as possible, we redetermined all titers using the same methods for all of these samples. And then we conducted a hypothesis-free genome-wide association study on these titers. This is just to show you that we put some thought into how to run the analysis, and we matched the samples from Sweden and from Germany so that they differed as little as possible in the potential confounders we knew about. And then we analyzed all the binding antibodies in Munich, and the neutralizing antibodies were analyzed partly in Innsbruck and partly in Copenhagen, but with a more or less the same method. And the split into Innsbruck and Copenhagen was randomized. And before analysis, and actually before any titration also, we split our samples into a discovery and a replication sample so that we could validate our own findings. The binding antibodies, as said, they were determined by ELISA in Munich, about 2,750 samples in total. And the neutralizing antibodies, they were determined using a luciferase-based reporter-gene assay. As a third phenotype, we also had a non-quantitative phenotype, the presence of antibodies. So this was any samples that were positive in the screening and then showed a titer larger than 0. When looking at the binding and the neutralizing measurements, we saw that they were strongly correlated. So binding antibody titers were both correlated with the neutralizing antibody titers by about 71%, but also with the presence of neutralizing antibodies independent of the actual titer. And this kind of correlation we can also see on the genetic level. So we can determine the SNP-based heritability of these traits. So that means we estimate how much of the variance of these titers is explained by the SNPs we have available in our samples. And with this approach, we find that the binding antibody titers has a heritability of about 36%, and for the neutralizing antibodies, it's a very comparable level. Interestingly, if we look at the neutralizing antibodies in the patients that already have antibodies, the heritability, the estimated heritability is lower and it's not significant anymore. Then if we compare again the binding and the neutralizing antibodies, we see that the genetic correlation is very high. It's 86% between binding and neutralizing antibodies, so that means that approximately by 86% the genetic factors overlap that contribute to the development of both of these. And a few more technical things before I show you some results. So the SNPs, based for this analysis, they were genotyped on Illumina microarrays, and then they were imputed, leading to about 8.5 million SNPs to analyze in both data sets. And we analyze these imputed SNPs, so these are actually then probabilities for a genotype and not the hard genotypes anymore. We also imputed HLA alleles. We had, for a subset of the patients, we had sequenced HLA alleles, and we could use this to confirm that our imputation worked well. As covariates for the analysis, we used treatment type, the duration of the treatment, and 8 components, like principal components, of the genetic similarity matrix to control for population structure. We did our analysis for the quantitative phenotypes with linear regression, for the dichotomous phenotype using logistic regression, and for the linear regression, one prerequisite is a normal distribution of the residuals, and to achieve this better, we transformed these titers so that they better follow the normal distribution. And now an important point is that we analyzed the Stockholm and Munich cohorts separately and then combined the results using a meta-analysis, because as you can see in this plot, the structure, the population structure, or in their genetic similarity matrix, so this is basically something like a principal component analysis of the genetic similarity matrix, you can see that there's systematic differences across the genome between these two populations, Germans and Swedish. So if we analyze them just together, there would have been the danger of false positive results, and this is why they were analyzed separately. But now to some results. So here you have a Manhattan plot for the GWAS of the binding antibody titer. And of course, as it's an immune-related phenotype, the Manhattan only has one high tower here. And what you can see in the x-axis is the positions of the SNPs across the genome, and on the y-axis it's the minus log 10 p-value of the association strength of the different SNPs with the binding antibody titer. And of course this tower is within the MHC region, and if we zoom into this, we can see a number of correlated SNPs that surpass this red line, which is the genome-wide significance threshold, which is typically a p-value of 5 × 10⁻⁸ in such genetic analysis. Now, of course, we had to prioritize, so this is based on the discovery data set with about 2,000 samples. We had to prioritize which of these signals to carry forward to the replication phase. And we basically decided to take signals that were not correlated with each other, that showed a very low linkage disequilibrium, and that remained significant when conditioning for the other signal. And this gave us 2 SNPs here, the ones with the red diamonds, that were then prioritized for the validation step. And one of these 2 SNPs clearly replicated, the other didn't. So this is the interesting one, the one that replicated in this analysis of then 750 samples, and it showed good support in both the discovery and the replication stage. Now, this selection and replication is based first on only the discovery samples. Now, it's no, let me first point out just this, that all of the, so this genome-wide analysis was run in linear regression using standard parameters, but because we could still have some deviations from the prerequisites for linear regression, like heteroscedasticity or non-normal residuals, all of these prerequisites were confirmed, and we confirmed these associations with permutations and so on. I'm not showing this every time, but we did it. So now, this is again the Manhattan plot for the discovery analysis. And if we pool the results from the discovery and the replication phase, this changes a little bit. And here the red diamond, this one here, is the SNP that showed the strongest support in the discovery phase and that replicated. But now in this pooled analysis of both, we actually see that another SNP a little bit downstream, the green one here, that has actually, this shows the strongest signal now. And therefore we also want to look at this one. So it's correlated, of course, with the SNP that was the top SNP in the discovery and that replicated. But we now want to know that we have a replication of the signal. We want to know like which one might be the most important or the causal variant. So there, this SNP showing the overall strongest signal is a candidate for that. And it has very good support in all 4 subsamples, discovery and replication from the 2 sites. And when we condition for this one in the overall sample, then nothing else remains significant. So this is, so to say, a good candidate of the overall strongest association. And here you can see a more detailed plot of this locus. So this SNP is located just upstream of HLA-DQA1. And in color you can see here the linkage disequilibrium, the correlation between the different SNPs. So you can see that it's like a whole block of associated SNPs here just upstream of HLA-DQA1, and this here again is the genome-wide significance line. So everything that is above this is significant. When further characterizing these 2 SNPs, so the first one here, the one that showed the highest association in the discovery alone, the second one showing the highest association in both discovery and replication combined. We have the summary again that both are intergenic, both are to some degree correlated with HLA alleles. They're both significant EQTLs, so expression quantitative trait loci. So the genotype of this SNP is associated with expression levels of transcripts of HLA-DRB1 in the case of the first SNP, and HLA-DRB1, DRB6, and DQA1 in case of the second SNP. And we determined these EQTLs in a set of German multiple sclerosis patients that were therapy naive. And both loci, this is just again some statistical background, so received strong support in both genotype and imputed data. So these are the SNPs. Now we'll move on to the HLA alleles. Here we see that 6 HLA alleles are significantly associated in the discovery stage and replicated in the replication cohort. In total, we analyzed 83 alleles after quality control that had a certain minimum allele frequency and so on. And what is interesting here about these 6 alleles that we found to be associated is that they might not be like completely independent, but what we actually have is 2 well-known HLA haplotype blocks. So there's these extended blocks of HLA alleles that originally come from ancestral populations thousands of years ago from Africa, and where some ancestral antecedents or ancestors of ours probably carried these kind of allele combinations. So what is interesting, so we have these two sets of haplotypes, the DR3 DQ2 haplotype that is has often been described as implicated in autoimmune disorders, and the DR15 DQ6 extended haplotype that appear to be associated here. And actually, I mean, this is still, we have to confirm this with further analysis, but looking at these two SNPs here, it is a possibility that they again are proxies for these two extended haplotypes. Now the next question was whether there are any treatment-specific effects. So the analysis so far contained the samples across treatments with treatment as a covariate, and then we also conducted the same analysis again, but per treatment group separately. We did not find any significant results for Avonex. We found significant results for Rebif, but they were the same as for the overall sample, so the same SNPs that I've already presented. And for betaferon, interestingly, we found independent risk factors, so one SNP and one HLA allele that were genome-wide significant for betaferon, but that were not significant for any of the other two treatments or for the overall group, including all treatments. This SNP here shows only like weak LD with the SNP that was strongest in the pooled analysis, and basically no LD with the SNP that was most significant in the discovery phase using all treatments. And once again, this SNP is part of EQTLs, and we have to analyze a little further to see like what might be the specific role of this SNP. But in addition to this SNP, we can also see that one HLA allele, DQA133, is significantly associated only in the betaferon treated patients and not in the ones receiving any other treatment. Okay, so this was all binding antibodies, and in the end, let me just compare this to the neutralizing antibodies. I've already pointed out that both titers were highly related and the genetic correlation was very high. And you basically see the same again here in these two plots. So here in both plots you have plots of the binding antibody titer, and in color you see the presence of neutralizing antibodies in these same samples. And here, especially on the right, you can see that there is a tendency that people with a high binding antibody titer then also had neutralizing antibodies, and people with a low binding antibody titers did not show them. But there's a very strong overlap between both groups here in the center. So this already hints in the direction that the binding antibody titers that we determined are not very well suited for as a proxy for the presence of neutralizing antibodies. We did the same kind of analysis in GWAS for the neutralizing antibodies, and a bit surprisingly for me, but very nice, is that the top SNP for both the neutralizing antibody titer and the presence of neutralizing antibodies is exactly the same SNP that was also the top SNP in the pooled analysis for the binding antibody titer. So it's really the same genetic factor that influences the development of these of both binding and neutralizing antibodies, which was not completely clear before. When looking at the HLA alleles for neutralizing antibody titers, we see a subset of the alleles that were significant for the binding antibody titers, and it's exactly the subset that is part of this, that forms this DR3 DQ2 haplotype. So these are, you can see by the frequencies, they're strongly correlated. And for the neutralizing antibody status, we could not find any significant HLA alleles after correction for multiple testing. which just shows that the so to say the power using the quantitative phenotypes was higher than using this the logistic regression based on a based on the screening and the presence of the of antibodies in the titration. As the very last thing, I want to explore the question: is it now likely several genetic factors that contribute to the development of these antibodies? So do we have a polygenic trait here where different genetic factors add up to influence the phenotype, or is it more something like a monogenic contribution? And we explored this using polygenic risk scores. So in polygenic risk scores, you basically take effect sizes from a training data set, from a GWAS, and then you go in your test set and you sum the number of risk alleles you find for a SNP, and you multiply it with the effect size from the training data set. So it's kind of a cumulative weighted score for the overall risk burden for this trait or disorder in your test set. And that then gives you for each individual an estimate of the genetic burden for the trait in question. And here I used, as training data, I used the GWAS from the discovery stage, and as test data, the replication samples. And looking at one of these polygenic risk scores, we can see that there is a significant prediction of, in this case, the binding antibody titer polygenic risk score for prediction of the presence of neutralizing antibodies. So you can see the people that have a higher risk score for binding antibody titer also tend to be more positive. The looking at the, so to say, prediction quality, we see an R-square correlation coefficient here of about 10% in the Munich samples and 25% in the Stockholm samples. That is pretty good, as it seems, as the overall heritability is about 34%. So this explains about 25% then of this. But what is a little on the other hand, if you just, instead of several SNPs, just take the top associated SNP, you approximately get the same R-squares. So it seems, although, I mean, we get a prediction with this polygenic risk score, it's not better than just using the single top SNP. And the same is the case when looking at the neutralizing antibody risk score instead of the binding antibody risk score. So it seems just taking the top SNP already gives us the best possible prediction in this case. And anyway, looking at this and looking at the strong overlap, we see the problem that we always have with these kind of analysis. Such a prediction might make sense like on a population level, that you, for example, look at the whatever 1% of people with the highest risk score and the 1% with the lowest risk score. But based on these analyses, I do not expect that we will have a prediction of neutralizing antibodies solely based on genetic factors. That will clearly not work. So to sum all of this up, we have measured binding and neutralizing antibodies. They're highly correlated, and the same genetic factors contribute to the development of both. It's mainly this one SNP I pointed out, and these DR3, DQ2, and DR15, DQ6 haplotypes. We could, contrary to previous studies, not find any evidence for risk factors outside the HLA region, so those were probably false positives. And trying to explain the heterogeneity of previous studies, it seems that part of this heterogeneity is due to different populations that were analyzed in the different studies, and there are clear population-specific frequency differences and also differences in the risk factors per populations already between Swedish and German. And then us having found treatment-specific SNPs also points in the direction that sometimes differences in the treatment within the different previous studies also contributed to the heterogeneous results. And the polygenic risk score analysis point in the direction that it's will probably be enough to carry on like a single proxy of the genetic risk into further prediction models, but not a complex genetic model based on several SNPs, at least when talking about the additive genetic risk. So of course, in addition to that, there could be more complex influences and interaction between SNPs, for example, that adds to this risk. And as a final point, let me just point out that for SNPs or HLA alleles with a frequency below 10%, we are still a bit, the power is still not optimal. So I cannot rule out that there's further low frequency risk factors that we could just not capture here that could not get significant in this setting of 2,000 discovery and 750 replication samples. This is all. Thank you for listening, and thanks to obviously many people at different locations that contributed to this.